All right, so I'm completing the video that is on chapters 19 and 20, BIO182, due to an interruption in internet service. I'm not sure why it was so bad. I was able to reestablish a quick internet connection at this point, but I sent out an announcement that we would cancel class and then I would record the remainder of the material and post a link. So I'm going to share my screen and we'll try not, I tend to talk a little bit too much when I do this, so I'm going to try to do it faster today. So one of the things that I've been told over and over again is that students will not watch long videos, so I'm trying to do this fast. So one of the things that I decided to do is, hold on, let me make this view present. Okay, so you guys are seeing that. And is that I'm going to hand off the Hardy-Weinberg equations to Khan Academy, and they do a great job. There's also Bozeman Science has a great video on Hardy-Weinberg. There's actually tons of videos online about Hardy-Weinberg equation. So in this, chapter 19 was on the evolution of population. So looking at how the DNA, which is broken up into sequences called genes, which code for proteins, how the alleles, which are the types of flavors of the gene, how the frequencies of those change over time. And that is evolution. So evolution is the change in the genetic structure of a population over successive generations. And so the genes themselves, it's still code for, say, the same trait like eye color, but the frequency of the alleles may change over time. And there may even be mutations for, say, new eye color that change. So that is evolution. So evolution is not just new species creation. That's not synonymous. Evolution is the change in the genetic structure of a population over time. So that's population genetics. Again, the Hardy-Weinberg equilibrium, I could spend a lot of time going over that, but it's actually done very well by Khan Academy. The equation of interest, this might remind you of a squared plus 2ab plus b squared equals 1. From algebra, it's a common equation, but in this case, we are looking at it applied to the frequency of alleles in a population. So the small lowercase p represents the allelic frequency of the dominant allele. In this case, represented by A. So if you have p squared, that's going to be the frequency of 2, p times p squared is 2. So that's going to be equivalent to the homozygous dominant frequency and similar to the homozygous recessive frequency. So I'm not going to belabor this. I said I wouldn't, so I'm going to try to just stick to that. Direct you to the excellent video from Khan Academy. So open in the new window. And so it's going to start off telling you about allelic frequency, and then the Hardy-Weinberg equation, and then applying the Hardy-Weinberg equation, and then discussions of conditions for the Hardy-Weinberg. And then down here, there is practice. And so I really want you guys to practice these questions. They'll set you up for success on your quiz. In addition, it can help you with the critical thinking, okay? And you can actually see the, solution. Okay, so again, I said I wouldn't spend all my time on that, so I'm not going to. We're going to go back. Don't want to do that. Oh. And why can't I go back anymore? right, so I'm not sure why I can't do that. So I'm going to go back into our course this way. 182, going back into our module 2, into our resources, and into the slides. All right, so here we go. View, let me just walk you through these. So Hardy-Weinberg, critical thinking question, you can answer those and upload those. Okay, so genetic drift. So we introduced this topic in the last module, and there was actually a video that I put into the resources by the Amoeba sisters, particularly on genetic drift. This is a commonly misunderstood idea. Genetic drift is a random chance situation, so that you just have a set of alleles in a population and randomly some are passed on. And because generally of a scenario like a low population size, the next subsequent generations may not be identical to the previous ones. So the change that you're seeing is called genetic drift. And so that change is due to random chance. So here's an example. Example is the first generation you start off with, you've got brown rabbits with allelic frequency of 0.5. So 50% of the alleles are brown and 50% of the alleles are white, this lowercase b. And then you have 10 rabbits in this example. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So you've got 8 of them are brown, 2 are white, right? Now we go to the next. So in this example, when the rabbits mate, you are going to see these rabbits survive up here, and they all happen to be brown. Now they're homozygous dominant brown and heterozygous, but there are no white rabbits that survive to breed. So in the second generation, only half of the individuals reproduce, resulting in a second generation with a change in the allelic frequency. So the change with P and Q, and the values are 0.7, which means that now 70% of the alleles are brown and 30% of the alleles are white. So we see a reduced percentage in the white allele. And so that's the genetics. The phenotypic or the physical expression, you still have a white rabbit over here because that's heterozygous recessive. You got one of those and the rest of the rabbits are brown. Now, if only 2 of these rabbits survive, so say there's very little food, just randomly, only 2 of these guys make it and they survive the coyotes and everything out there, then the final generation that we're looking at, the third generation, they're all brown in this scenario. And that was just by chance. It wasn't because the white rabbits were very visible to the coyotes, or the white rabbits got skin cancer because their brown fur didn't protect them from the sun. It was not because of the environment. It was just random chance. And that's what genetic drift is. So in this case, we end up in third generation, and we only have the allele that is the brown allele. So P is 1. That means 100% of the alleles are the dominant brown allele, and Q is 0. So 0% have the white allele, the lowercase b. Okay, so that's genetic drift. Genetic drift is just a change over time due to chance. Okay. And so this is just a summary slide of that. So you started off with equal distribution of brown and white alleles. And then because there was only a small n equals 10 rabbits to begin with, and even a fraction of that is reproducing, then you're getting this pretty pronounced change. And that is common when you have very small populations. Okay. And yeah. This is just showing the change in P and Q and the relative amounts. So this is just an interesting graph. Don't worry if you can't understand it or interpret it. It's not gonna, you're not gonna fail in the class. It's basically just saying that if you take the red and the blue, the sum of these, the sum of the red and the blue and the green are going to add up to 1 here. Okay. All right. Okay, so a type of genetic drift. Genetic drift is kind of an umbrella term for random chance changing the frequency of DNAs. It's random chance change is a driver of evolution. A specific example of genetic evolution is called the bottleneck effect. And that's just where you have a big population and you just take a few of those that survive. So if you have a catastrophic event and just a couple individuals survive, those individuals are going to change whatever they have. It's going to represent the allelic frequency of this new population. And that may be very different than the previous. So in this case, there's some yellow original individuals, whatever this yellow represents. Those guys were not, they did not survive. And so the surviving population now only has blue and white. All right, gene flow. So this is where populations may be fairly isolated, but then occasionally you do have an introduction of new genes. So that may be if you have, in this example, I think you have beetles that hang out on the grass and beetles that hang out in the soil. The grass beetles are green, the soil beetles are brown, but on that edge zone, you're having some gene flow. And so occasionally you got a brown beetle going into the grass and mating, and that can, that change, randomly a new beetle comes in, can have an effect on the allelic frequency in the population. So this again is, it is sort of random, just that random beetle walking over there, and it is an example of genetic drift. Okay, so however, genes are not the only scenario that is responsible for phenotypic expression of traits. And one example is an environmental factor. So you could even consider this like epigenetics. So it's something outside of the genes themselves that contribute to the expression of the gene. So this is the case of the American alligator, Alligator mississippianus. And so sex is dependent on temperature in these guys. So eggs incubated at 30 degrees C produce females, eggs incubated at 33 degrees C produce males. And this is a type of a sexual selection that is seen in alligators and turtles and many different reptiles. So although we're talking about phenotype being directed by genes, it's just a point of interest that it can also be affected by other environmental factors. Okay, all right. Now we're going to talk about different types of selection. So for different types of selection, you can have a type of selection in the environment, meaning selective force, that results in the physical expression of a trait keeping about the same frequency of expression over time. That's called stabilizing. So example here, selection, an average phenotype is favored. So an average physical expression of a trait. So in this case, the physical trait that is expressed is the number of eggs laid. So robins can lay just a couple eggs. They might lay 3, 4 eggs, 5 eggs, 6 eggs, 7 eggs, but they typically lay 4. And the reason for this is there's a selective pressure for, it's disadvantageous to lay more eggs. Larger clutches may result in malnourished chicks. So if you lay more eggs, your overall health of your chicks are going to suffer. And then if you have a very small clutch, then maybe you have 0 offspring. So in the end, this is the example of stabilizing selection. So the intermediate or the middle phenotype is reinforced generation after generation after generation. So please know this example or another example of stabilizing selection for the quiz. There's also directional selection. And so in this case, you may have heard of it. It's the peppered moths. It's where there is the environment changes and there's a new selective pressure introduced because of the environmental change. And that means that a new phenotype is favored. So in this example, the original population of the peppered moth in England was on trees, and the trees were often had white bark in the surrounding environment, and they blended in pretty well. However, during the Industrial Revolution, there was a lot of coal and soot production that basically coated everything in black. And so the white moths were very easily picked off. And so in the original population, although the majority of the moths were peppered colored, there were some moths that were black. And once the soot covered everything, the black moths they survived, and so they reproduced. And so this directional change, where over time you see a decrease in the allelic frequency of this pepper color and an increase in the allelic frequency of the black color, that's called a directional shift, okay, or directional selection. And then after the Industrial Revolution, once there became the awareness of measures and the ability to produce more clean fuel, there was a shift back toward that original population. So that's directional selection. And then there's diversifying selection. And in diversifying selection, so you're starting here with the white rabbit in the middle, so an intermediate phenotype, and then you're seeing over time a shift towards 2 phenotypes that are not like the middle one. So it's diverging into two phenotypes. In this case, a rabbit that is partially white and black, and a rabbit that is basically all black, okay? And so in this scenario, you have this hypothetical population. Gray and Himalayan rabbits are better able to blend in with a rocky environment than white rabbits, resulting in diversifying selection. So that means if you're white and you are born in this rocky environment, you're at a disadvantage. But if you are either of these other two, you have equally advantageous fur. So that's a diversifying population. So they're both have a higher fitness compared to the white rabbit. Okay. So the critical thinking question associated with this is looking at populations. So you pick one of these populations. The first one is brown trout. So brown trout populations that are exposed to extensive fishing by humans. What effect would that have on that original population? And you can think about any phenotype you're interested in. One that's commonly studied is size. So the size of the fish. How is fishing going to change the average maximum size of the trout? Something like that. Is it going to select for small fish or medium fish or large fish, this extensive fishing? And then similarly, you can think about longhorn ram populations exposed to extensive hunting. What effect would hunting have on longhorn rams? Are they all hunted equally? Are there certain rams that are prized more than others? And if those are culled from the herd through hunting, what effect will that have on the resulting phenotype in the population. Okay, so both of these are actual scenarios that are occurring. I would, since you're doing this outside of class, just ask you to think about it on your own before you look up the answer. There's no right or wrong answer, and you don't have to look up the answer. I would actually encourage you not to, and just think about it on your own. But just what would be the effect of humans on these populations, and pick one or the other, and then just talk about how you might see whatever phenotype it is of interest change for those animals that are fished or hunted. Okay, so that's critical thinking question 2. And there's just the 2, I think, questions in this. Let me look real fast. Yeah, that's about it. Wow, we got a lot to go over. Okay. So that's the second question. Okay. Frequency dependent selection. So this is actually a lizard that's local here in Arizona. So a yellow-throated side-blotched lizard is smaller than either the blue-throated or the orange-throated males, and appears a bit like the female of the species, allowing it to sneak in copulations. So in this case, this is just an example of a lizard that it's really small. And so you think, well, why would the small lizard be successful? Shouldn't it just be the big lizards that dominate and impress the females? In this case, they can kind of sneak past the males and get in there and copulate. So there is a selection for the big males that are going to impress the females and, you know, fight each other. And then there's the small males that can just sneak in there past the big males and get in and copulate and get out. That's actually seen in quite a few types of organisms, fish, lizards, monkeys, those type of things. Okay, so sexual dimorphism, this is just a term that refers to the fact that in many species, males and females have different characteristics. They look differently. So the male in the bird species is often very flamboyant or more colorful than the female to impress the females in mating rituals, and also it's hypothesized for the females to blend in and camouflage, especially when they're sitting on their brood, their eggs. Spiders, very common. The females are much larger than the males, and they often live, you know, many more years than the males. It's interesting in terms of just the physical expression, but also like the lifespan of a lot of organisms is different in the males and females. And I might ask you, what do you think the difference is in humans? Is there a difference? And there is. One sex is definitely longer lived than the other. And then, so ducks, there again, birds are always the go-to for sexual dimorphism. Okay, so that's chapter 19. Chapter 20, the history of life. So we're going to look at, in this chapter, more the relationships, the evolutionary relationships between organisms. And so remember, a phylogenetic tree is a mechanism to diagram the evolutionary relationships of organisms. So we'll look at phylogenetic trees and just what the diversity of life is. In this semester, we're going to go into a lot of detail in subsequent chapters. So starting next chapter, where we go into viruses, and then we go into bacteria, and then we go into fungi and plants and animals. So we're going to look at all of the diversity. But first, we're just going to give an overview of how we create diagrams to represent that diversity and what that diversity kind of looks like. So there's over 2 million species cataloged currently. Total number of species varies depending upon your estimate, 5 plus or minus 3 million. So, and total number of individuals, 100 billion, a trillion. That's going to be those multicellular organisms. If you start talking about bacteria, you've got, you know, over 1 trillion bacteria in your body. So lot of diversity out there. Okay, so types of phylogenetic trees. So phylogenetic tree, you have on the left here what is probably more common that you're familiar with, the rooted phylogenetic tree. You start off with a common ancestor at the root. That means that all of the life that you see on the ends here as you go up, all have the common ancestor represented at this point. And so time t equals 0, at least represented here, is at the bottom, and then you're going up. You have a speciation event, and you have species A and species B, and then you have multiple speciation events as you go on, creating many different lineages. The three largest groups, if you remember from BIO181, the three domains are bacteria and archaea. Both of these are unicellular prokaryotes, so they're only one cell big, and they're called prokaryotic. That means they don't have a nucleus or other organelles. The third domain is eukarya. These are both unicellular. So important note, some eukarya are unicellular and multicellular. So there's both in this domain, and it includes plants and fungi and animals, but often the overlooked protists or protista, and those are actually very abundant. Anything that does not fit into the animal, fungi, plant category gets lumped into protista. They are generally aquatic and single cellular. All right, so that was just a review of overall three big buckets. Instead of looking at it with this root on the bottom, now most phylogenetic trees, the root's in the middle, and this is partly just for conservation of space. It's much easier. We've got tons of species that we know about to date. So you can just get a lot more information if you do this unrooted tree in the middle, and it represents the same information here. Okay. General vocabulary. So taxon is just it's a nice word if you don't know if you're talking about genus or family or genus or family or class or phylum. You just say taxa, and taxa is like, okay, we're talking about a group, and that's in general. A clade is a branch or lineage, so you're talking about a evolutionarily related group, and again, that's a general term. So the root of the tree on the bottom here, and then you have branch points. At each branch point, you have what you can refer to as a clade. The branch point is going to be a common ancestor for everything that's distal to that on that tree, or further out from that. Okay, you need to memorize these again. Hopefully you memorized them in 181, maybe not. If not, you're going to memorize them here. This is the classification system for taxonomy, and it's starting with the broadest domain, kingdom, phylum, class, order, family, genus, and species. Few mnemonics, ways to remember that are dumb kids play catch on freeways going south, or how I learned it was don't keep pouring cement on Fred's green shoes. Anyway, that's the order from small to large, or from large to small. Okay, so memorize those. And then as we go forward, let me just point out that the genus and the species, the last two classifications or taxa, those are what constitute the scientific name of an organism. So if I ask you for the scientific name, that is asking for the genus and the species. And so this example, oops, over here on the right, the species is Canis lupus. That's going to include dogs and wolves. So Canis lupus, the genus is Canis, and then the species here is Canis lupus. Kind of a hard one because Canis is in there twice, but so the genus is Canis lupus, the species is Canis lupus. So it's Canis lupus. All right. Okay, so homologous we learned this in the previous chapter, but I'm going to ask you to know it again. Homologous, same evolutionary origin. So this image here is showing the bone structure in the limbs of animals, human, dog, bird, whale. It has the same structure, one big bone, then two bones, then a whole bunch of little bones, one big bone, two bones, a whole bunch of little bones, one bone, two big bones, a whole bunch of little bones, even into the whale. The whale is a mammal. It was a quadruped that was land dwelling before its ancestor went to the ocean. So it did not evolve in the ocean. It evolved as a land dwelling animal, which is why that you can see it has the structure, which is very different from other organisms that evolved in the ocean, say like the shark. Okay. And then you have analogous. So analogous are structures that have a different evolutionary origin. And so that was mentioned in the review today. The wing of the bird or bat is a different origin than the wing of an insect. They do not have a common winged ancestor. So you cannot trace that wing back to a common ancestor. They both evolved it independently as a mechanism that conferred flight, and that is advantageous for a number of reasons, right? To get out of predators' way, to access new food, to move to different environments. So that shape of that, it's basically that foil kind of shape to push and propel air, in this case, evolved independently in both of these lineages. All right. So that's an analogous structure. So here, and you may see evolutionary reversal, which means that some characteristics that are evolved are lost later on. So like a snake, if you look at the snake, it has these tiny little legs. Its common ancestor for the snake, its ancestor was a quadruped, and a snake lost the legs. So if you look at a snake and a worm and you just say, oh, they must be same ancestor, that's not always true. Sometimes there's what's called evolutionary reversal. And so organisms that look the same, again, a worm and a snake can both move through their environment without these kind of appendages that might get in the way. So they're functionally useful. That's an example of analogous structures. So similar analogous characters may cause taxa to appear related when in fact they are not, or only distantly related. So which two of these animals are more closely related? Shark, dolphin, cheetah. So it turns out the dolphin and the cheetah are both mammals, so they both give live birth and produce milk. They have a lot of common characteristics. They have a common ancestor that is more recent than the shark, which evolved many millennia ago in the ocean. Okay, so even though the dolphin and the shark look considerably closer in appearance, they actually are not closely related when comparing them to the dolphin and the cheetah. So please know that example as well. All right, so molecular systematics. So now that we have molecular data, it's changed everything. We're getting a much better, more precise, a clean picture of relationships between organisms. It's evolving. The trees that we had before, a lot of them have been significantly revised and are still being revised as we get more information. So DNA and RNA, those are both nucleic acids, and by sequencing DNA, the genome of organisms, and then putting it into the computer and having the computer figure out how similar those DNA strands are among different species, phylogenetic trees are automatically generated using computer algorithms. Sometimes because there's only 4 different nucleotides, that means A, T, C, and G. Sometimes there are patterns that actually have like the same letter sequence, but that's just because they randomly have the same letter sequence. So that can happen. And then different types of mutations might also give sequences that look related. Sorry, if you have mutations or changes in the DNA, you might find that species appear like they have, they're not similar. They don't have as much similar DNA as you would expect because of the mutations and frame shifts and big insertions of chunks of DNA or moving DNA around, which is actually fairly common in many organisms over time. Okay, so why the heck is it important to study evolutionary relationships? Here's just a few ideas if you want to justify it. We can learn about what species we're related to, and since we can't really do very many experiments on humans, we can do experiments on them and figure out some information that might help humans. You can track evolution of parasites and viruses, which has been really important with COVID-19, trying to figure out where it came from, its origin in bats. It's interesting to note that bats are 30% of all mammals. So even though it might seem like, wow, how did it come from a bat to a human? Odds are pretty good. There are a lot of bats out there in the world, and they're mammals. And so the chance of a crossover between a bat virus and a human is actually pretty good, and probably has happened many times before and will happen in the future. So it's important to understand how that happens. Biotechnology, so by figuring out evolutionary relationships where can sequence organisms' genomes and then actually use that to create enhanced organisms, produce more effective drugs, improve conservation efforts. So this is kind of important by sequencing the genome in species and figuring out which are closely related. We can try to conserve a broader, more diverse section of species that represent a lot of variety versus ones that are closely related. Okay, a monophyletic group is just a group that includes all of the descendants of a given ancestor and includes the most recent common ancestor. So monophyletic group, so you might say the lancet, lamprey, fish, lizard, rabbit, and humans are all monophyletic. They're all vertebrates. Okay, so they're in a monophyletic group. All right. Again, evolution does not lead to perfection. It usually takes a lot of mutations to create functional phenotypic expression. So there's most mutations are not helpful, or what's called deleterious. They're not helpful. They result in non-functional genes or in not viable embryos. The rule of parsimony. So this is basically just saying that the simplest explanation is probably the most likely explanation. So if you are computer algorithms and you're trying to figure out which species are related, and there's a really complex explanation and there's a very simple explanation, you go with the simple explanation. It's kind of like Occam's razor, if you've heard of that. You go for the simplest option, and that's the governing principle as far as creating some of these computer algorithms and lineages. Okay, so species concept, we use the word species all the time. If you really want to get into a conversation with a biologist, you might end up defining the species concept. It's going to be different for a microbiologist as it is for, say, a large animal biologist, wildlife biologist. So biological species concept, different species have interbreeding populations, okay? species have interbreeding populations. So if you are a biological species, you can interbreed and have viable offspring. However, there's another species definition, which is the phylogenetic species concept. And this is species is a population or a set of populations characterized by one or more shared derived characters. So this is really going to be helpful if you're a microbiologist or if you're looking at organisms that don't pass down genes from parent to daughter cells or undergo sexual reproduction. So both are useful in their selected fields. So species traditionally, when you're talking about it, assumes vertical gene transfer. So that means you're passing down DNA from a mother cell to a daughter cell. So this would be the example in animals, right? However, it does not account for a horizontal gene transfer, which is the primary way that most genes are actually passed through organisms, because most organisms are prokaryotic. So horizontal gene transfer, which is abbreviated HGT, is the transfer of genetic material between unrelated species, all prokaryotes, some eukaryotes, endosymbiosis, reverse transcriptase. Okay, so this is, you can get horizontal gene transfer between every type of prokaryote does this. So bacteria and archaea can pick up DNA or genes from unrelated species. Their DNA is lying around in their environment. It is not uncommon for these unicellular organisms to bring it into their cell in a vacuole and potentially incorporate it into their genome. This is responsible, one of the mechanisms that leads to antibiotic resistance in hospitals. There's a lot of organisms in there, bacteria, that have antibiotic resistant genes. And if those genes are laying around, say after those bacteria die, other bacteria can just kind of suck those up and incorporate them into their genome potentially. And it's kind of an area where you can create super bugs because they're all able to pick up the genes that allow them to survive, which may be in species that aren't not related to them. Again, and it's hard when I use these words, the word species, because in bacteria, it's defined differently. Okay. All right. We talked about endosymbiosis in 181. That's where you have the creation of the mitochondria from a free living organism, the creation of the chloroplast from a free living organism. Eukaryotic cells do have, historically have engulfed and kept the DNA of other living organisms. It's not common, but it does happen enough to be important. And reverse transcriptase, this is actually a viral, it's an enzyme that allows viral DNA to be incorporated in our DNA. So humans have as much as 30% of our DNA is actually viral in nature, meaning a virus, we were infected with the virus had an enzyme, reverse transcriptase. That viral DNA was incorporated into our genome and then it just got stuck there. And so we have a lot of that. And it's debated how much, but estimates are about 30% that I've seen recently. Okay, horizontal gene transfer. So in bacteria, genes can be, please know these three mechanisms for horizontal gene transfer. One, transformation, that's when DNA that's lying around in the environment is just taken up. Two, transduction, genes are transferred by the virus, by a virus. So a virus can actually transfer genes to another organism. And then conjugation, where you have DNA that is transferred via a physical connection of the plasma membranes. There's a pilus here, which the DNA, this is shown on the right, this piece of DNA called a plasmid, this pink thing, is going to actually be replicated, and then a replicated version is going to be passed to this other bacteria over here. And these are all examples of horizontal gene transfer. So you're not having, think about mitosis or meiosis in animal or eukaryotic reproduction. You have a parent cell and the DNA is copied, and then you have daughter cells that are produced, and the parent cell turns into the daughter cells, either 2 exact copies or 4 different genetically distinct copies. In this case, you have a cell here, and it's not dividing, right? But it is changing its genome. So it's getting new genes from the environment without dividing. So it's not undergoing reproduction, but it's getting new genes. And this is horizontal gene transfer. So the first type, transformation, picks it up from the environment. Second type, transduction, transferred by a virus. So some sort of bacteriophage, which is the name of a virus that attacks the bacteria, comes in, dumps in the DNA, gets put into the genome. Or conjugation, where you have 2 bacteria, in this case, that are transferring just a piece of DNA between them. Okay. So you're not having splitting of the DNA or the cell. Okay, this does occur occasionally in eukaryotes. There's not a lot of evidence, but it's pretty interesting when it does happen. So for example, transpoons or jumping genes have been shown to transfer genes between rice and millet, which are different plants, and somehow the genes from the rice got into the millet, and we don't exactly know how. So in plants, there's evidence of this. In fungus, there's evidence of this. So there's evidence of, for example, the uptake of the taxol gene from the yew tree. So the fungus is living in certain environments on the yew tree, and it has been able to incorporate a gene that is in this plant cell. So it gets the gene from the plant cell, incorporates it into its own genome, and now as part of that fungus, when the fungus reproduces, it reproduces that same gene that was in the tree. And then the bottom here is carotenoid transfer. So this is what the picture is above. So a carotenoid is, if you might have heard that term before, it's a pigment. It's also involved in photosynthesis. I always think of it, it's kind of orange and has that root carrot in there, like carotene. So the carotenoid enzyme was transferred from a fungus to an aphid. So again, they're not the same type of organism, and somehow the gene got from the fungus into the aphid. Aphids are small organisms with a stylet that goes into the sap of a tree and exudes or pulls out from the phloem sugar. And over here is showing, so they're usually green, over here is showing that some of them indeed turned this orange color after obtaining this enzyme that produces this orange color. And the source for that was from a fungus. So pretty cool. Of course, we're doing this as humans all the time now, pulling genes from one species and putting it into another. So there is a precedent for this in nature. It's not as common. Humans are now doing this. It's called genetically modified organisms, and we're doing it all the time. Okay, endosymbiotic theory. That's just where the mitochondria and the chloroplasts were originated. How they arose was that you had engulfment of a prokaryotic cell by a larger proto-eukaryotic cell. So you had, originally everything was a single cell, everything was a prokaryote, and then over time you had eukaryotes evolved. So the idea of how eukaryotes evolved is you had a big eukaryotic, proto-eukaryotic cell. So before it was a eukaryotic cell, it engulfed another free living organism, and now they lived happily ever after. One of those free living organisms looked very similar to mitochondria does today. So that's the theory, the endosymbiotic theory. You can look at that slide for more details. Another example of that type of relationship, the endosymbiotic relationship, genome fusion. So this is basically how the nucleus originated. Also, for bacteria, the idea of how, and you don't need to memorize this, but there's a really big category of bacteria that are called gram-negative, and they actually have two plasma membranes. And so the way that they evolve two plasma membranes, the idea is from the engulfment of gram-positive bacteria by archaea, and then you have this combination here. So kind of like a hybridization. Endomembrane system, it's just an example there if you want to go back and look at how that originated. Okay, and then the last couple of slides here are just on alternatives to the phylogenetic tree that we're used to. So we're used to seeing, okay, we have one common ancestor of life, everything originated from that one cell. And, you know, there are alternatives to that view. And one of them is this web and network type of view where you have transfer of genes from very divergent species and lineages over time. And so it's not like a clear branch. So you have evolution and then you've got this horizontal gene transfer that's occurring. An example shown here, right? The chloroplast that is in cyanobacteria. The idea is that that cyanobacteria was engulfed over here, and then you had the origin of photosynthetic eukaryotes like plants and algae, right? So you're swapping the DNA. So it's not like in a tree you can just say, oh, everything starts from the bottom and you get these subtle changes. You're getting these big infusions of, in the case of a chloroplast, it has quite a large genome that's a circular genome in that organelle itself. So you're getting large infusions of genes to other lineages, other clades. So it's not clear as you'd like it to be. And especially in the origin and in the unicellular context where you have single-celled organisms, they're swapping genes all the time, left and right. And that's a big old mess, right? So that's kind of the bottom here. So there's this other idea, an alternative to the tree of life, the ring of life model. And so this may help explain how certain eukaryotic genes more resemble those of bacteria, while others more resemble archaea. So here in the middle, you still have T equals 0. So this is like the origin of life. And since we don't really know, we don't have evidence of these cells. There are not preserved fossils. They didn't have bones. And so this is largely theoretical. And so when you get back to 4 billion years ago, the idea is maybe there were, you know, quite a number of different unicellular organisms, there likely were, that were constantly swapping genes. And so it was more of like this pool of genes that were being transferred. And so that's represented by this circle. And then after some period of time, there became more clear individualized species, if you will, that were no longer interbreeding, and you got these like large categories, in this case the domains eukarya, bacteria, and archaea. So pretty interesting field. I think I would get a huge headache if I really tried to look into trying to reconstruct this, but there's a lot of really great scientists that are working on this, and we may never know, you know, what happened here initially, but we can try to work backward to the best of our ability. And we do know that there are genes that are from archaea that look like archaea that are in eukarya, and there are genes from bacteria that are in eukarya. So we do have some guideposts. Okay, so video on natural selection. This one, I put a whole bunch of videos, and this was still too long, so I apologize for the length, but I put a whole bunch of videos. No, that was my other class. I put one video into the audio video resources. I'm thinking about the virus one. Next week, the virus one, I have a lot of videos for. But this one is the types of natural selection. So
customize your car insurance with.
You can watch that.
You can manage your team's work easily with Monday.com.
And Khan Academy, I mentioned for the Hardy-Weinberg. Also for the Hardy-Weinberg, one thing that I was going to go over in lecture, so for Hardy-Weinberg equation to be accurate and applied, there are requirements. So if we look at requirements for Hardy-Weinberg equilibrium, I'm going to search for that. Let's see. I'm going to go to my table of contents, and we're in chapter, here we go. So one thing I just want you to go over is the requirements for Hardy-Weinberg equilibrium. Where is it? Where are the requirements? The requirements are basically theoretical, that you have an infinite population that's not under any selective pressure. And so that means you're not getting organisms moving in and out. Here we go. So if a population is in equilibrium, there are no evolutionary forces acting on it. So generation after generation, it has the same gene pool, genetic structure, and the equations hold true for all of time. However, right, even Hardy and Weinberg recognize that no natural population is immune to evolution. So populations in nature are constantly changing in genetic makeup due to drift, mutation, migration, and selection. So the only way to determine the exact distribution of phenotypes in a population is to go out and count them. So this is theoretical, right? So for this to occur, you would have a static population, and that is not the scenario. So, but it gives you an estimate, a snapshot in time, and it's a way to start, right? So you don't have to just say, oh, it's not perfect, we're not going to use it at all. It's an estimate, but just know that things are evolving over time, and so these frequencies will change. Okay, so that's it, and I will see you all next week when we will talk about viruses. And